Okay, well, we finished talking about protein synthesis, mRNA translation, and we looked at the regulation of translation, and we looked at regulation of messenger RNA stability, of post-transcriptional and post-translational regulation of gene expression. And basically what's happening now is we have the protein is synthesized, and it's in the cytoplasm, because of course the ribosomes are in the cytoplasm. And the idea here is that the proteins need to get to various places in the cell. So that's what we refer to as intracellular protein transport. That is, how does it get around from place, from place to place and to where it needs to get to. And it's also sometimes referred to as protein localization or protein sorting. So here we have a cross-section of a cell, and we're looking kind of at some of the things that are in the cell. Of course, we have the nucleus, shown in brown, surrounded by a double nuclear membrane. The outer nuclear membrane is continuous with the endoplasmic reticulum, which actually goes, it's a membrane structure that goes throughout the entire cell. And we're going to look at what the function of that is in a few minutes. And what happens is, of course, the RNA gets out, the messenger RNA gets out through the nuclear pores, and then it gets to the ribosomes where we translate proteins. And then those proteins need to get to various places, for example, the mitochondria, or back into the nucleus, or the Golgi apparatus, or they're secreted. In fact, this particular type of cell is a pancreatic cell. It's called an SNR cell, and these secrete digestive enzymes. And the way these cells are situated is at the bottom of the cell is attached to a kind of a matrix, and then all the secretion takes place at the top of the cell. So it's a unidirectional secretion, and that'll be important a little bit later. So there's two types of ribosomes that I want to just point out, and we're going to be talking about those as well. So there's what are called free ribosomes. These are floating around in the cytoplasm. And then there are ribosomes that are attached to the endoplasmic reticulum. And you can see those represented by the black dots. And these are referred to as rough endoplasmic reticulum. They look rough under a microscope because of the ribosomes that are attached to them. So what we're going to be looking at is how proteins, which are synthesized on these ribosomes, get to the locations they need to get to. So this kind of breaks it down in a kind of a simple way. And the title is Membrane Enclosed Organelles, which include the nucleus, the mitochondria. In plants, there would be the chloroplast, and then there's vesicles like the peroxisome, and there's other vesicles in the cell. There's an endosome, and they get in to these organelles by different mechanisms. So they have, for example, in the nucleus, they're transported through nuclear pores, and we'll look at that in a moment. Some of the organelles that do not have pores, they're transported actually across the membrane. So here it shows you proteins made in the cytoplasm. Somehow they're transferred across the membrane into the chloroplast, into the mitochondria, into the peroxisome, and then, or into the ER, and we'll talk about that as well. And then there's transport by vesicles. So if you look down towards the bottom, it shows the ER, the endoplasmic reticulum, transports proteins to the Golgi by means of vesicles, membrane-bound vesicles that fuse. So there's a few different ways for proteins to enter specific regions. And we're going to look particularly, we're going to give some examples. We're going to look at the nucleus, and we're also going to look at the endoplasmic reticulum. because those are kind of the two that have been, well, most well studied, and also they give good examples of the different mechanisms of through nuclear pores and across membranes. We'll also look at vesicle transport. Let me show you this next slide. So this is, of course, my rendition of this, and I call it protein sorting. And again, it represents proteins are made, on the ribosomes and the cytoplasm, and they have to get to all these different places. They stay in the cytoplasm, go to the Golgi, they get secreted, they go to the nucleus, they go to the mitochondria. And as it turns out, there are two distinct pathways for protein sorting. There's the cytoplasmic pathway, which is on the left-hand side. And through the cytoplasmic pathway, that refers to cytoplasmic ribosomes. Ribosomes that are free in the cytoplasm. They send proteins to the mitochondria or the nucleus or the chloroplasts. But then there's another pathway which actually leads to proteins being secreted or going into membranes. And these are carried out by membrane-bound ribosomes, which represent the rough endoplasmic reticulum. So what this is showing here, and we'll look at that in more detail in a few minutes, either secreted from the cell or some membrane-bound protein, or proteins that need to get into the ER or the Golgi. They go through a very specific pathway, through the endoplasmic reticulum, into the Golgi by means of vesicles, and then they either get stuck in the membrane or they get secreted. Now, either of these pathways and any location that a protein needs to go is dictated by what are called localization or signal sequences. These are amino acid sequences that are part of the protein that dictate where the protein needs to go. Generally, they end up binding proteins which help target the protein, the protein of interest, to where it needs to get to. So let me give you an example of some of those. So here's a table taken from the book, and again, signal sequences. These are amino acid sequences that are part of the protein. They direct the proteins to the correct compartment. So there's just a whole bunch of examples here. So for example, import into the ER. This is and it shows the amino acid sequence on the right-hand side. So if these sequences are present on a protein, they will end up getting imported into the ER. Then there is imported to the mitochondria. So here you have a series of amino acid sequences, and these sequences, if it's present on a protein, that will target it to the mitochondria. And then of course it's similar to import into the nucleus. These are called nuclear localization signals. So if you have proline, proline, lysine, 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 arginine, lysine, valine, you put it on any protein, it'll end up in the nucleus. So these are amino acid sequences that target the protein to a specific location. Now, a couple things I want to say about that. If you look at, for example, the signal sequence for import into the ER, that has to cross the ER membrane. And many of those amino acids in that signal sequence, the ER signal sequence, are nonpolar. If you look at all the leucines, valine, glycine, many of these are nonpolar, and that actually participates in helping it through the membrane, because of course the internal part of the membrane is nonpolar. So it helps it get through. That's one characteristic. Now, the other point I want to make is, again, I'm going back to the information content in the DNA. Now, many of these signal sequences are removed after the protein gets to its location. So if you import into the peroxisome, that sequence is removed. If you import into the ER, that sequence is removed. Import into the mitochondria, that sequence is removed. So the sequence, the amino acid sequence that's there, is not part of the functional activity of the protein. It's specifically there for targeting it to its correct location. And that, again, is encoded in the DNA. So it's not just the function, but it's also the location is encoded by the nucleic acid sequence. I want to start out by looking at the cytoplasmic mechanism, and that represented the nucleus and the mitochondria and the chloroplast. We're just going to look at the nucleus. So this is the cytoplasmic pathway. So it's actually rather, in a sense, straightforward. Nuclear proteins contain this nuclear localization signal that's part of the protein, recognized by nuclear import receptors. So on the left-hand side, it's showing the nuclear pore, which we've kind of looked at before. And the top part is the cytoplasm. Of course, the proteins are synthesized in the cytoplasm. And then this is to import into the nucleus. So what kind of proteins would need to be imported into the nucleus? DNA polymerase, single-strand binding proteins, topoisomerase, histones, telomerase, all the splicing proteins of the spliceosome, all those need to be synthesized in the cytoplasm and end up back in the nucleus. So they all have nuclear localization sequences. What happens is the protein is synthesized, and that's kind of represented in green, has a nuclear localization sequence, and it's bound by a particular nuclear import receptor. And that has a specific name, which we'll talk about in 1 second, in the right-hand side. But basically, the idea is it binds to a particular receptor, and that complex, which contains the protein destined to go into the nucleus and the nuclear import receptor, is then brought to the nuclear pore. It's brought to the nuclear pore quite often by motor proteins attached to microtubules. So they're transported to that location. And then they bind on the surface by, they call them cytosolic fibrils. Sometimes they're referred to as nucleoporins. These are receptors that bind to the nuclear import receptor and bring it to the surface. And it brings it to the surface, and it allows it to enter the nucleus through the nuclear pore. It's an active process. It requires energy. So on the right-hand side, a little bit more details how proteins are imported into the nucleus. So you have, number one, a nuclear localization signal on some particular protein. Let's say it's telomerase. And that needs to bind to this nuclear import receptor, and that receptor is referred to as important. Important is a protein, which is the receptor. It's bound to important, and then that binds to the surface of the nuclear pore by these various nucleoporins or additional receptors. It's brought into the nucleus, and so in part number 2, which is on the bottom, it says the protein important complex enters the nucleus and binds to another protein in there, which is called RAN-GTP. It's another protein bound to GTP. And what happens when that complex binds to the protein important complex, the protein that's destined to get into the nucleus is dissociated from that complex, but the important remains bound to the RAN-GTP complex. And so the protein is basically now entered the nucleus. Now, the RAN-GTP complex is allowed to leave the nucleus. The RAN protein actually has a nuclear export sequence. It gets out of the nucleus, and then in step number 4, the important is released, and the GTP is hydrolyzed. That's the energy consumption, and both of them move to the cytoplasm it says GTP is hydrolyzed, important is dissociated, and then it's allowed to recycle and bind to another protein with a nuclear localization sequence. So it's a very active process of getting these things into the nucleus. So I'll just say similar processes happen in the mitochondria and the chloroplast. They bind to particular receptors on the cell surface, and they're allowed to enter the organelle, the mitochondria, or the chloroplast. But I want to talk a little bit more about the other side, the right-hand side, which is a secreted protein or membrane-bound proteins, because they have a different mechanism of entering these organelles, like the endoplasmic reticulum. It's a process called cotranslational transfer, which I'll describe in a few minutes. But let's just look at a protein that's being secreted. So here we're going to be looking at the pathway that a secreted protein takes as it exits the cell. And what it says here is it enters the pathway through the endoplasmic reticulum, and this is an experiment that kind of demonstrates that. And we're using those ACNR cells. Those are pancreatic cells. It's spelled ACINAR. And the power of these particular cells, while they're used as a good model system for this, is that they secrete from one side of the cell. So it's a specific orientation. And so you can watch them go out the top. Watch the proteins go out the top. Now, the way the experiment is done is by using pulse chase labeling. And we talked about pulse chase labeling when we looked at DNA replication and the replication bubbles. Same principle, but of course, here we're looking at protein synthesis. So rather than use a radioactive nucleotide, we end up using a radioactive amino acid. And in this case, it's called tritiated leucine, which is radioactive hydrogen and a leucine amino acid. So the way this experiment works is you have these cells growing, and they're oriented in a specific direction, and you add to the media, to the growth media, radioactive leucine, and you add it for 3 minutes. After 3 minutes, you wash out all the radiation, all the radioactive leucine, and you replace it with non-radioactive leucine. So the first 3 minutes is the pulse, so pulse for 3 minutes, and then you have the chase. The chase is for, in this case, 17 minutes. So original 3-minute labeling, and then 17 minutes without labeling, and then again 3 minutes labeling, and then 2 hours without labeling. And so at the end of that time, you then go ahead and fix the cells, and then you expose them to either digital detector, which can detect the radiation, or sometimes people will use X-ray film. You can put it over the dish. That's called autoradiography. And then you can visualize the cells, and you can visualize where the radioactive protein is. So any protein that was synthesized is taken up and incorporated the radioactive leucine. So at 3 minutes, and if you look at the cell, they have some things that are identified. So you have the rough ER, the Golgi complex, then there's secretory vesicles, and then there's the extracellular space. And again, we're following the pathway of the protein. So in the first 3-minute pulse, when you look at that, you see the dots, those black dots. Those black dots represent where the proteins are during that 3 minutes. So what you see is that newly synthesized proteins, they're being synthesized on ribosomes, but they are somehow associated with the endoplasmic reticulum. Now, when you do the chase for 17 minutes, what happens is the proteins that were originally synthesized during that first 3 minutes are labeled, and then after 17 minutes, we can see where have they gone. And what we see is after 17 minutes, they're up towards the top of the ER, all those black dots, and they're actually entering some vesicles, and they're clearly entering the Golgi bodies, Golgi complex. And then after an additional time, after 2 hours, what you see is they have left. There's no radioactivity in the ER. There's still radiation and radioactivity in the Golgi, but now you have a lot in the vesicles, and in fact you can see some up at the top fusing with the cell membrane and being released to the extracellular space. So the conclusion from this experiment is the pathway of proteins that are secreted. So I have that written on the right-hand side. It starts out with ER-bound membranes, then you have movement into the ER, then you have movement into the Golgi bodies, then you get the passage of the proteins into the secretory vesicles, and then you get fusion of the secretory vesicles with the plasma membrane that then allows all the contents out. That's secretion. We're going to focus for a moment on the rough ER, what happens there. Because the proteins that are on the ribosomes getting synthesized, how do they get into the ER? So this is just an electron micrograph of the ER, and I just wanted to see what it looks like because we always draw it with these little dots, and in fact when you look under the electron microscope, that's what you see. So proteins are synthesized on the outside of the ER. So the ER is a double membrane structure. So you have membrane on one side, membrane on the other side is like a tube, and on either side is the cytoplasm, and on the inside it's called the lumen. So the lumen of the ER. And the proteins need to get from the surface bound to ribosomes into the lumen of the ER. And the way that happens is through a process called co-translational transfer. And the implication just by that word is that they're transferred as they're being translated. So the first thing to note is that there is one pool of ribosomal subunits. Remember, the ribosomal subunits are separate, and then during the initiation process, they come together and make the full ribosome. So the small and the large are separate. That's what's shown in the middle. Now, on the top, it's showing the free ribosome cycle, and that's pretty standard, pretty much what we looked at before. You get the initiation, the protein, the messenger RNA is bound. You get the proteins bound to be synthesized, and you see the polyribosome and the cytosol. Proteins coming off, and that may be a protein that stays in the cytoplasm, or maybe a protein that ends up in the nucleus. But these are occurring on free ribosomes in the cytoplasm. Now the other pathway is what we're going to be talking about, is the membrane-bound ribosomes, and these are the rough endoplasmic reticulum. And what we're seeing here in this little diagram, we'll look at it in a little bit more detail in a moment, is that the synthesis begins on a free ribosome. You can see that on the left-hand side. And then that little red thing coming out, that's the ER signal sequence. So this is what signals the protein, that the protein that's destined to be synthesized is destined to enter the ER. And that protein then makes it over that ribosome, with the protein being synthesized, makes it over to a receptor, which we'll look at in a moment, and then the ribosome locks onto that receptor, and then the protein is synthesized and forced through a pore that is part of the receptor complex. And so you can see that happening. They call it a protein translocator. But as synthesis is occurring, the protein is being pushed into the lumen of the ER. So this is referred to as co-translational transfer. So let's look at a little bit more detail. So here we see some of the steps more at the molecular level. So if we look on the left-hand side, we see a ribosome translating a messenger RNA. It's making a protein. It's the growing peptide chain, and the ER signal sequence is shown in red. Now, as soon as that ER signal sequence is exposed from the ribosome, it goes through kind of like a tunnel, and it takes a certain number of amino acids. I can't remember how many, but maybe something on the order of 20 before it starts to appear at the surface of the ribosome. But the first thing, because the signal sequence for the ER signal sequence is at the amino terminus, that's the first thing to be exposed. And so when that thing is exposed, it is then bound by a receptor. And that receptor is called the signal recognition particle. Signal recognition particle binds to the ER signal sequence. Now, when it binds to the ER signal sequence, it inhibits translation. So translation is halted. And the reason why it's halted is because we don't want this protein synthesized in the cytoplasm. We want it synthesized in association with the ER membrane. So it halts or inhibits protein synthesis until the signal recognition particle, which is in brown, interacts with a signal recognition particle receptor. And that's shown towards the middle. There's kind of that dark blue, half moon type thing. So SRP receptor binds to the SRP, the signal recognition particle, so it's brought that ribosome to the surface of the ER. Now, that signal recognition particle is released, but in the process, there's a transfer of the ribosome with the associated protein to this additional protein complex called the protein translocator. And so it kind of moves over. The signal recognition particle moves it and attaches the ribosome. Actually, the ribosome interacts and links to the protein translocator. And at that point, the protein synthesis then resumes. And now it's resuming. And that green represents the protein. And you can see that as the protein is being synthesized, it's being pushed into the lumen of the ER. Now notice that that red little part, which is the signal recognition sequence, the ER signal sequence, that remains associated with the protein translocator, and it actually gets removed by an enzyme called signal peptidase. So let's look at a little bit more in the next slide. Now this is showing really kind of what happens as the protein enters the endoplasmic reticulum lumen. It's showing it without the ribosomes. So what you see on the left-hand side is kind of where we left off before, that you have the protein being synthesized. It's being forced through the protein translocator, but the ER signal sequence remains associated with that, but the rest of the protein goes through. And then there's an enzyme called signal peptidase, and that's shown in yellow. It actually cleaves the protein and removes the signal sequence from the protein. Once it's in the ER lumen, it's not needed anymore, so it's cut off. So that's what I was saying, that these things are present not for function, but only for localization. So signal peptidase cuts off the signal sequence. So often that's kind of left as a remnant in the membrane. Sometimes it's degraded. But ultimately, the entire protein gets, without the signal sequence, gets pushed into the lumen of the ER. Now, when we talked about proteins earlier, one of the things I said was that these proteins fold up spontaneously on their own, which is true. But it's not unusual for the proteins to misfold. And so there are additional proteins which are called chaperone proteins, which are present in the ER lumen. So these are, one of them is called BIP, BIP. And so these are chaperone proteins that assist in proper folding. Now, before I say anything about that would be a protein that started out with co-translational transfer in the same way, that it had a signal sequence, it got in there. So if I were to ask you on a test, for example, which of the following proteins do you think had an ER signal sequence? And one of them was, this BIP protein, that would be a candidate, right? That's the only way it could have got in there. And there's other proteins we'll talk about later. So the chaperone proteins, it says here, it assists in proper folding. It assists in a very special way. I mean, it can't know what the protein's supposed to look like. So what happens is the protein folds spontaneously on its own. The green folds spontaneously on its own. And if it's all folded up properly, that's all fine and good. If it's not folded up properly, then the chaperone protein will denature it and let it start again. How does it know it's not the proper folding? It knows it's not the proper folding because there are numerous, or I'd say excess, hydrophobic amino acids, nonpolar amino acids on the surface. That's one way. So if you have all these nonpolar amino acids on the surface, there's something wrong. They should be on the interior. And so it'll bind to it, denatures it, and lets it go again. It doesn't guide the folding. Okay, so that is co-translational transfer. That's how proteins get into the ER. Now these are proteins that can stay in the ER, like the BIP protein, for example, or they go through that pathway that we talked about, which would be the Golgi, and then ultimately secretion. So let's look at that pathway. So what we're interested in is the secretion portion. I'm not really going to talk about endocytosis. I'll just say with endocytosis that there's proteins that would bind to the plasma membrane on the cell surface, and then get taken in by the formation of a vesicle, then gets transferred to a lysosome, which often is used to degrade the proteins. So that's the pathway in. But we're looking at the secretory pathway. And the way the pathway works is that the flow goes from the left to the right. So it shows the ER. We now know how proteins get into the ER lumen by the co-translational transfer. And then they pass through the Golgi. They pass through the Golgi. First of all, they need to get from the ER to the Golgi, and they do so by these transport vesicles. So the proteins are in the ER, and then they get captured in these vesicles as they bud off of the membrane, and then they get targeted to the Golgi. So you can see these things going into the Golgi, and then they get passed through the Golgi by vesicles, again by transport vesicles. So you can see from one part of the Golgi, it gets captured in a budding part of a membrane, which becomes a vesicle, then it reattaches, proteins enter, and that cycle goes on. Now there's proteins that stay in the Golgi. So they have particular retention sequences that allow it to stay in different parts of the Golgi. And we're going to look at the Golgi in more detail because they have specific names. But this is just to illustrate you the pathway. And then finally, at the top of the Golgi, which is referred to as the trans Golgi, the cis Golgi, there's cis, medial, and trans. The cis Golgi is closest to the ER. The medial Golgi is those three parts in the center. and the trans Golgi is up on the right-hand side. But in any case, what happens is the proteins that are destined to be secreted, which they call that exocytosis, is they get, again, put into transport vesicles. Those vesicles are a little bit different. They're clathrin coated, means they're coated with a protein. They're coated with a protein that makes them very stable and allows them to interact with the interior surface of the plasma membrane, and you can see that on the bottom. And then the red arrow showing that it ultimately fuses to the plasma membrane. So the plasma membrane kind of grows in a sense, but what was inside that vesicle then gets released. So I just want to show you what the clathrin-coated vesicles look like electron microscopy. So these are pictures of secretory vesicles, basically. They're transport vesicles, but they're very specific ones. They're secretory vesicles because these are clathrin coated, and you can see that it's on the left-hand side. It's budding off of the Golgi, and you have these proteins that are attached to the inner surface as well as the outer surface to help create this vesicle. So they're very well structured. But and on the interior, in the interior of that vesicle are the molecules, the proteins that are going to be secreted. So secretory vesicles are coated with this protein, clathrin. So the secretory pathway is kind of like a default pathway. What do I mean by that? In order to get into the ER, there's clearly ER signal sequence. But then there are what are called retention sequences. If you look at that chart at the beginning, there were a few retention sequences listed. So there's the ER retention sequence. That means the protein gets into the ER and it stays in the ER, like the BIP or the chaperone proteins. If it doesn't have a retention sequence, then it gets put into a transport vesicle, and then you can see it moves up, fuses with the Golgi. And I have it illustrated here. The cis Golgi is the first one closest to the ER, then the medial Golgi is in the middle, and then trans Golgi. But the proteins move by these transport vesicles to the different levels of the Golgi, the cis, medial, and trans. Now there are specific proteins in each one. So there are cis Golgi retention sequences, there are medial Golgi retention sequences, trans Golgi retention sequences. Clathrin, for example, would be a trans Golgi retention. It would have a trans Golgi retention sequence because it's there to help build a clathrin-coated vesicle. So proteins will stay there. In the absence of any additional retention sequences, then the default is that it gets packaged into the clathrin-coated secretory vesicle, and you can see it makes its way up to the membrane, and then it fuses with the plasma membrane, and the contents are released. On the right-hand side, again, it just shows electron micrograph of the ER, and then it shows the Golgi. Looks like kind of like layers of pancakes. One additional thing that I'll point out, which we'll talk about later, are the cell surface proteins. You can see those. I have them drawn kind of as a Y. Those are proteins that are embedded in the membrane, and they get embedded in the ER membrane. You can see them there, and they get packaged into the secretory vesicles, and they move up also. And then because they're embedded in the membrane, when the secretory vesicles fuse, when the membrane fuses, then those things remain in the membrane and they're pointed outwards. So the Y is shaping up, pointing up, and that is supposed to represent some kind of a receptor. Could be a receptor for a growth hormone, for example. So any kind of cell surface protein that's bound to the surface is embedded in the membrane, and it goes that way. And later, I'll talk about how those get put there. Okay, so I want to kind of finish up in a way, just talking about the Golgi. Because what happens in the Golgi is quite significant. What happens is glycosylation. Glycosylation is the addition of sugars. So complex sugars are added to the proteins. Actually, it starts out in the ER with a addition of a series of sugars, and then ends up going through the cis, medial, and trans Golgi, where the sugars get modified. So I'll say this, and I'll probably say it again, every single protein that's either on the cell surface or in the ER or in the Golgi or secreted, like a hormone that's secreted or a growth factor that's secreted, is glycosylated. Sugars are added. So this just represents a little bit more detailed image of the Golgi. So you have the cis Golgi on the top. That, of course, is closest to the ER. And then you have, they call it cis cisterna, media cisterna, trans cisterna. I don't go into all those. I just call that whole cluster there the medial Golgi. So the medial Golgi would represent those three together for me. So we have the cis at the top, the medial towards the center, and the trans Golgi is what's closest to the plasma membrane, and the secretory vesicles butt off of that. Now there's two things that happen in the Golgi. One is glycosylation, which we talked about. All proteins that pass through there get glycosylated. And then there's also what's called proteolytic processing. That also occurs in the Golgi. We'll talk about proteolytic processing right at the very end. So just very briefly about glycosylation. I'll tell you don't need to know the names of the different types of sugars that will be mentioned in this one and in the next slide. They have glucose, mannose, acetylglucosamine. These are sugars that get added in the lumen of the ER. So this shows you got co-translational transfer going on, and then they're usually attached to asparagines, particular amino acids. And so you have in the membrane, you have a protein bound which has this linked, it's called lipid-linked oligosaccharide. And that oligosaccharide gets cleaved and moved over, and it gets attached to the asparagine. And so you can see on the right-hand side, there's a sugar there. Every asparagine, well, I don't know about every, but many of the asparagines get glycosylated. And you can see the sugars are complex. Glucose is a sugar on its own. Mannose is a sugar. And acetylglucosamine is a sugar. So you can see these are highly linked and complicated structures that get added. And then what happens when it moves into the Golgi, it gets modified. So here it's, glycosylation is modified in the Golgi. So it takes place in the ER, that's shown on the left-hand side. And you get the asparagine is modified by the complex sugar that was added. And then as it goes through the Golgi, through the cis Golgi, the medial Golgi, the trans Golgi, different things happen. And so what you see is the addition of, for example, high mannose gets added. It's a particular type of sugar, and then you get this N-glucosamine transferase transfers another sugar to there, And so the bottom line is that the sugars get modified. And at each level of the Golgi, they get modified in a different way. So every protein that goes through the pathway gets glycosylated. The surface of the cell is completely covered with proteins. There's receptors, there's transporters, we'll talk about those later. There are pores, there are cell-cell proteins that are required for cell-cell interaction. These are all glycosylated. So one question is, what's that about? So the entire surface of the cell, which is covered with proteins, is actually glycosylated. So you have the proteins there, and then you have this series of sugars that are kind of making, you can imagine it as a halo around the cell. Now it turns out that these sugars do not affect function at all. So they're not allosteric or anything like that. They don't change the structure of the protein. They don't change the function of the protein. The thought is that they protect the protein, or they protect the cell surface proteins from proteases that may be in the environment. So it seems to be serving as a protective mechanism for the outside of the cell, particularly the proteins on the outside of the cell. Now, the last thing I want to mention is that proteins that pass through the Golgi, many proteins, get what we call proteolytically processed. And then that proteolytic processing activates the protein. For example, many hormones get proteolytically, they're made in an inactive form. And that in order to activate them, they have to get cut. Some part of it has to be released so they're processed. A good example are digestive enzymes. So digestive enzymes are processed and activated as they're leaving the cell. So it's like you don't want to have an active protease or digestive enzyme chewing up the interior of the ER or chewing up the interior of the Golgi. So they're inactive until they are processed by a protease that cuts at a specific point. So let's give you one example. So the example here is the proteolytic processing to generate glucagon. Glucagon is a hormone that is involved in controlling blood sugar levels. And so it's synthesized on the ribosome, and of course this protein would have had as part of the protein, it would have had an ER signal sequence at the amino terminus. That was cut off. That's how it got into the whole system. Got moved up through the system, through the Golgi, and when it reaches the trans Golgi, well, it's synthesized as an inactive precursor, which is called proglucagon. It's a 37 amino acid protein, but it's not active glucagon. It's proglucagon. And then there's an enzyme called convertase. Convertase is an enzyme that exists in the trans Golgi. So this, again, would be an example of a protein, convertase, which as it started out, had an ER signal sequence. That's how it got in there. But it also had a trans Golgi retention sequence. But in any case, the convertase has a very specific amino acid sequence that it cuts. So the convertase cuts the proglucagon, leaves a little bit off the end that gets degraded, and then the 29 amino acid peptide or protein is the active glucagon, and that of course will fold up appropriately, and then it would get released from the cell by the fusion of the clathrin-coated vesicle that it would be in. Okay, so that's overview of how some proteins get to where they need to get. They all have to have specific targeting sequences or signal sequences. And we looked a little bit of detail at the nuclear localization sequence, and then we looked a little bit more at the sequences, particularly it would be the ER signal sequence that gets the protein into the ER by co-translational transfer, and then all the various activities that take place as it moves from the ER to the Golgi and out of the cell. Okay, you have a good rest of your day.